So last speaker before lunch, and it gives me great pleasure to announce Zachary Hunter, who I've got to know as a brilliant researcher, but also as a very nice person, very modest, even though he did receive the Robert Kyle Award, which is a great honor for him. And I must say, on a personal note, I was really impressed with you because I took my children to the opening ceremonies on Thursday, and they were really touched by what you said during your acceptance speech. So that's a very special to me. Zachary Hunter.
Thank you so much. It's really a pleasure to be here and to be able to talk about some of the work and some of the amazing things that are going on in Waldenstroms right now. Some of which is from our group, but a lot of which, for those of you who, well, if you could have been at the meeting earlier, it's really an incredibly inspiring time. The research is moving at a pace that I would not have previously thought possible. And many of it actually thanks to people who are here and who you'll be hearing from throughout the day. So I'm more of a basic scientist, not a clinician, so I'm going to talk a little bit about some of the new genomic findings in Waldenstroms. I'm going to start off actually by talking about really the basic science. And before anyone gets worried, it's going to be very nice and gentle. I'm going to try, but feel free to come and ask me questions later. So I work at the Dana-Farber Cancer Institute with Steve Treon at the Bing Center for Waldenstroms Macroglobulinemia in Boston. And again, we're splitting the talk into two parts. One focused on just trying to understand when we talk about genetics, what does that mean? We talk about DNA, RNA, protein, why do we study these things? And then in the second half, talk about what are the findings in WM and how are we taking this from an abstract scientific idea, which someone like myself might get very excited about, to actual things that matter in the clinic, which is really the only thing that matters in the end, is how can we take some of these ideas, move them into the clinic, and improve lives of patients. So what is DNA? DNA is, in fact, just made out of 4 basic pieces, and these pieces are designed so that they actually pair together. It's A, G, C, and T is our sort of shorthand for the different types of components of DNA, and as you can see, the G's goes with C's, the A's with T's, and it forms this very, very nice double-stranded complementary molecule. And this is incredibly stable. And this is actually what gets passed on from parents to children. And so all of the ways that you go, I just have a daughter actually, 7 months, and so everyone's always like, oh, she just looks like you. 7 months, it's kind of hard to say, but but that's DNA, right? That is information being passed on from parents to children across the generations. So what is it used for in the cell? DNA is used as a template to tell your body how, and your cells in particular, how to make things, how to run. And it sort of serves as a master copy of how to do everything. And if you think about, let's say you were to go and you were to build a beautiful church like this, it would take years and years and years and years. And you'd have someone, an architect, who came up with a master set of blueprints. Those blueprints, there are tons of craftspeople that would be necessary to build an organ, for instance, or to make a chandelier, or work on the ceiling or the basic architecture. And you can't have everybody using that same set of blueprints. Not to mention, the organist doesn't really need to know that much about the details of the columns. So what people do is they make copies of that blueprint. This is the parts that each individual workman needs to be able to do their job. And that's exactly what happens in the cell. The DNA is the master copy, but it makes no sense to have it run all over the cell trying to get this work done. So copies are made of just pieces of it. So simple sets of instructions, and that's RNA. So DNA gets copied into a single-stranded molecule called RNA, and that goes out into the cell and actually does the work. So to review, you have DNA, it serves as a template, gets transcribed into RNA, and then that RNA actually encodes for how to make proteins. So I know people have heard about proteins. Proteins are what do a lot of the work in the cells. They're the receptors, they are motors, they move the cells around. And this is sort of how, at least when I was in school, how this was essentially taught. Now, DNA makes people nervous, right? This idea that there is this sort of fundamental program that is controlling everything. And it can feel deterministic. And for a long time, that's how people thought about it. And I don't think that's quite the right way to think about it. And I think that in the scientific community in general, people are moving away from this idea. And one of the things is that when they've done twin studies, actually, by the time people are much older, they actually look very different from each other. They have very different lives. And yet they have the exact, identical twins have the exact same set of DNA. So clearly this is not 100% deterministic. And we're going to talk about that. But the main thing I want people to get out of this is that DNA is a blueprint for building proteins and not a blueprint for who you are. So any anxiety you have about this, let it go. It's okay. But one thing about DNA is that there's a lot of it. So there are about 3 billion base pairs in the human genome. And this is actually from the Sanger Institute in England. And this is a print edition that they made. And actually, if you look closely, it's not even the whole thing. That's a lot of information. Now, what's really incredible is that amount of molecular information is packed into every single cell in your body. Not only that, you actually get that 3 billion base pairs from both of your parents. You get one copy from each parent. So all that information is crammed into this little tiny nucleus in the center of every single cell in your body. And if that doesn't blow your mind, I don't know what will. It's really a truly amazing thing. And this does cause problems, right? It's so long, you could actually, if it was thick enough, you would actually be able to see it if you strung it out end to end. So because it's so large, it actually isn't accessible. And this actually sort of should answer a lot of questions that you might have if you start thinking about it. If the DNA is the same in every single cell in your body, why is your blood different, respond differently to your skin, to your liver, to your heart? All these different tissues act very, very differently from each other, even when given the exact same response. And that's because effectively they don't see the same DNA. There's so much of it, it's so tightly wound up that actually only little pieces of it are available at any given time. And so the DNA that each tissue receives or is able to see or utilize is slightly different. And this is something that we call epigenetic alteration. Which parts of the DNA have been unwound and are accessible, and which ones are tightly bound up. So for the first time, just in the last decade or so, we can actually go and start to measure these in real ways, not just look at individual genes at the DNA level or the RNA level, look at different ways in which the DNA is being modulated physically. But we can actually go and we can see a genome. We can look at all the RNA in a cell, known as the transcriptome. We still can't look at all the proteins, but we can look at a lot of proteins. And we can start to try to put this together. But as you can imagine, this is really quite a lot of work. Now, if you look here, people used to teach about this as going like DNA, RNA, protein, and it's just very one-way process. And now that we understand a little bit more about epigenetics, we know that the proteins go and they modify the DNA. Not in a permanent fashion, but again, controlling which aspects can be used, which can be seen. They make chemical annotations on the DNA about which aspects should be used. And so this actually winds up being a dynamic process. So really, what we're talking about is an interaction with the environment. And this is entirely dynamic. And again, you can think about this as, think about the DNA as being your operating system on your computer. I'm willing to bet that almost everybody here who owns a computer either uses Mac OS or Windows, right? The DNA in your computers are all the same. And I bet you none of you use your computers in exactly the same way, right? It's true. Like the operating system is going to tell you the limits of what you can and can't do. But within those limits, there's quite a bit of variability. And this is really how we're starting to understand what's going on in cells. However, in any individual part of this is really quite complicated. So this is actually just one whole genome sequencing result from a patient, very sort of top-level view. And so now these days, people like Ramon Garcia-Sanz, like myself, like people actually all over the world are trying to take information like this, pair it with additional information like from the RNA, from the protein level, to try to understand what is unique about Waldenstroms patients and what we can use with that information to develop new therapies. So I'd like to transition and talk a little bit about the genomics of WM. And again, don't focus too much on the individual genes. There's a couple which are important, and the rest of the names, you can just try to get the major concepts. But before we go there, I want to talk just a little bit about mutation in cancer. So mutations are changes in the DNA. So it's an amazing thing that your cells, when they divide, can copy all these 3 billion base pairs pretty well, actually. I mean, I can't copy several hundred things without making a mistake, let alone 3 billion. But occasionally, through chemical alterations, through exposure to ultraviolet light, to just random happenstance, mistakes are made. And many of these mistakes don't matter. It's not a big deal. But some of them do. And as they accumulate, the cell's programs start to change. Certain proteins don't work anymore, or they work in ways that they were never intended to work, and gain new functions. And eventually, this can cause cells to forget what they're supposed to do and what they are, and they'll start to grow out of control. And when that happens, we call it cancer. So in Waldenstroms, the most common DNA misspelling, or common mutation, is this mutation in MYD88, which you heard Ramon talk about so nicely. And we see it in over 90% of these of WM patients. And what is MYD88? It's a gene that is actually used to help your body detect viruses and bacteria. It attaches to receptors that are looking for these things and will actually come together and help activate downstream signaling to the cell when those things are detected. And what happens with the mutation is it allows this protein to activate without bacteria and viruses actually being present. And this causes your immune system to get really excited, right? Hey, I saw something, we need to fight this. This is really important. And unfortunately, with all the other mutations together, this causes the cell to grow and survive. Now, this data has actually been now observed all over the world. And as Ramon said, roughly in sort of the same basic rate, somewhere around or just over 90% of WM patients. And we can also see it in that precursor condition that Dr. Kyle described, IGM-MGUS, in, again, about 50%. So Guang Yang, who works with me, actually right across from me, was working on this, and this was this classical pathway that had always been described, but it wasn't working for him in terms of explaining Waldenstroms. He could inhibit MYD88, he could prevent its activation, and you can see these little green dots that are there in the blue circles here. This is this protein called NF-kappa B, and it gets activated, and it helps these cells grow and survive, and we've known about it for a while, but we didn't know why it was activated. And he was able to show that when he blocked MYD88, he could get all these green dots to go outside of this blue area. And this blue area is the nucleus where all the DNA is. So if you could get this NF-kappa away from the DNA, you could really shut these cells down, and actually it would result in them dying, ultimately. So it was very exciting, but when he tried to cut it off a little bit lower down, it didn't work. Or it didn't work as well as we would have expected. And so this got him looking for other ways of explaining the phenomenon. And one of the things he discovered is that there's this molecule called BTK that was also able to do this exact same process, but was independent of these other proteins called IRACs. And this is important because we have drugs against BTK. So if you've ever heard of the drug ibrutinib, this is silencing this molecule right here. So we had seen just this one mutation in MYD88, which you'll occasionally hear referred to as L265P. But in other diseases, in fact, there's a whole bunch of mutations that are observed. But it's so frequent in WM that, in fact, we often will just test for this one particular mutation and nothing else. And one of the things that both our group and others have started to observe is, well, they're rare in WM. These are the ones that we had initially reported. We have started to see some of these other ones as well. And so they do exist. They're not common, but we are really encouraging clinicians in particular that when, if you get tested and it shows that you don't have this mutation, it's probably worthwhile trying to find out whether or not some of these other mutations might possibly exist. As far as we can tell, they have very similar effects. The other thing I'd like to talk to you about are mutations in this other gene called CXCR4. So when we first noticed these sets of mutations, we noticed two things about them. One, that they're all located here at the end of the protein, right, and nowhere else. And the second thing is that there's different types of mutations, and some of them change the spelling of the protein, and some of them just shut it off, right? And these were more similar to a combination of ones that actually scrambled it or shut it off. And those are the only ones that we'd see, and they're always right here. And why was this important? This is actually sort of a picture of the three-dimensional structure. And the signal, that's the part that sticks outside of the cell, is right up here, and this is the internal part that signals. What we're missing is this area in yellow. And it turns out it's not actually all that important for signaling. A little important, not terribly important. What it is important for is turning it off. So what happens is when this gets activated under normal circumstances, you can almost think of the cell, you can see these almost like little handles, and the cell is actually identifying like, hey, this is turned on, we got to get this thing turned off again, let's grab a hold of it and let's pull it back inside the cell. Unfortunately, in these patients, this has been cut off, and so it's no longer there, and so that it can still signal, but it can't get internalized. So it'll sit on the cell surface, and it just kind of keeps going and going and going. And when we put these two things together, one of the things that we noticed is that all the people who had these CXCR4 mutations also had MYD88 mutations. And so we could start breaking patients up into three different categories. Those who had MYD88 mutations but no CXCR4 mutations, those patients who had both mutations, and those people who had neither mutation. And this resulted in us being able to start to understand a little bit about what we already knew about the disease. We just didn't know why. So people who have neither mutation actually, when they first present, at least in our clinic, typically have lower levels of bone marrow involvement and lower levels of serum IgM. Whereas those people who have, especially these truncating mutations in CXCR4, had the highest levels of both. We also noticed that the people with these truncating mutations came in with slightly more symptoms, and that all of them together, any kind of CXCR4 mutation, had very low rates of adenopathy. And in the laboratory at least, cells with these mutations displayed a lot of resistance to chemotherapy. Though I want to stress, there's a very big difference between what we do in the lab and what we observe in patients. I think one of my favorite comments when I first started working here was from Linda Pilarski, who pointed out that we've actually been curing cancer in mice for years. Now, one place where we have seen the CXCR4 demonstrate a big effect is actually in ibrutinib. So when we conducted this trial of the BTK inhibitor ibrutinib, we noticed that those people who were mutated for MYD88 but did not have the CXCR4 mutation, they had these very deep, beautiful responses, very large increases in their hemoglobin. And those people who were missing both mutations, however, did not respond well, did not have the same increase in their hemoglobin. But those people who had a combination of both mutations were somewhere in between. They responded well, but maybe not as well as those with MYD88. And so this has definitely inspired us to try to figure out how can we help improve things for this group of patients. Though I do want to point out that the responses are still quite good for them. Now, we've only talked about MYD88 and CXCR4, but I just want to show this slide to say that there's a lot more going on, and that here in this meeting there are people talking about additional mutations and talking about how we can start to integrate this information into WM to discover more pathways, more therapeutic targets. But at this point, I want to actually switch gears a little bit and talk about the transcriptome, the RNA that's coming off the DNA. So we just finished a large study of 57 patients, and we're now trying to make sense of it all. You can see we were able to associate genes with lots of different aspects of WM, and I'm just going to give you a few of the highlights that I think are maybe important. So we were not the first people by any stretch of the imagination to show this, but we were able to observe big changes in this gene family known as BCL2. Now, I don't expect anyone to remember that, but the thing to know about the BCL2 family is this controls a lot about cell survival. So whether when cells get sick or they get old, they have this program within them to be like, you know what, it's time to go, make room for a new cell. And that is all controlled by a balance of these sort of pro-death and pro-life proteins that are part of this family. And one of the things we noticed is that the pro-life, pro-survival proteins the RNA at least was really quite high in the Waldenstroms patients, and some of the pro-death proteins were unexpectedly low. And one of the things that's good about this is that there are now inhibitors that are just entering clinical trial against BCL2, and hopefully in the next couple years we will actually have really good data about how this works in Waldenstroms. The other thing I wanted to point out is, we talked a lot about these mutations in CXCR4, but we actually found that CXCR4 itself and a number of its associated proteins, such as VCAM1 and CXCL12, were really overexpressed in these Waldenstrom cells in comparison to the healthy donor B cells that we analyzed. And again, this sort of gives us some hope that maybe, maybe we can target CXCR4. We can use it as a therapeutic target, and maybe this will help improve things in Waldenstroms. There's actually also a clinical trial now in development for CXCR4, and actually here at the meeting, a number of people proposed novel research projects at this point, looking into ways of exploiting CXCR4 for therapeutic purposes. So again, coming hopefully in the near future, we will have some good data on this, though this is all still preclinical. The main thing I also wanted to say is that, I talked about these three different groups of patients, those who were mutated for MYD88 and wild type for CXCR4, those who had both mutations and those who had none. Each group had a very distinct expression profile, and that's really all I want to point out here. Is just simply that you can see that there are really a large number of genes that are really unique to each group, and this means that we can start to think about targeted therapies and also personalized medicine, trying to figure out, as we move forward, what therapies are going to be best for each individual patient based on the mutations that are present in their tumors. We're not there yet, but for the first time, this is something that we can actually talk about, and we can actually do prospective trials keeping track of this and start to measure, like, okay, are there people who do well? Are there certain, does this predispose people to certain symptoms? Is there anything we can do about this? And so it's a very exciting time. Lastly, I want to talk just a little bit about a program that we did actually in conjunction with the Netherlands. And we did some machine learning, which is just a fancy way of saying we had the computer do all the heavy work, to try to understand what genes were involved in bone marrow involvement. And we got a number of genes that we expected, but also a new gene, CXCR4, which we'd never really looked at before. And you can see here, CXCR4 and MYD88 both played prominent roles as well. So Josephine Vos was visiting us for 6 months and was actually able to go back and look in the serum of patients with Waldenstroms macroglobulinemia and show, in fact, that the CXCR4 levels correlated beautifully, both with the amount of bone marrow involvement and also with the levels of hemoglobin in patients. She's now followed this up, looking at how cytokines change with ibrutinib, and it actually really looks like CXCR4 may eventually, it's still a research project, but may eventually become a really great biomarker for this disease. And its function is something that we're still trying to understand, but again, will hopefully open up some new exciting therapeutics. And as Guy Sherwood mentioned, I know Dr. Kirsten and Dr. Pauls just won a really, really exciting and prestigious grant from the IWMF. So I just want to highlight there's some awesome research happening here that you guys should be really, really, really proud of. So in conclusion, this is sort of a basic overview of CXCR4 and MYD88 signaling. We barely understand it, so I definitely don't expect any of you to understand it. But what is important is that we're now starting to be able to rationally go through and make targeted therapy. So we now have ibrutinib that is approved in the US and here in the EU, and that targets BTK, but there's new BTK inhibitors that are coming down the pike, which is really exciting. So hopefully there will be new alternatives. Maybe this will also make it a little bit more affordable. We'll see. Venetoclax, our ABC199, is that BCL2 inhibitor that targets cell survival. That's in clinical trial right now in Waldenstroms. So we will actually learn about this soon. And then there's a trial opening soon for ulacupamab, which targets CXCR4. And as I mentioned, also there are a number of sort of more experimental projects which are going on right now in Germany and throughout the world, trying to figure out new ways of targeting this to see if we can improve the lives of patients with WM for you, which is why we do what we do. And again, these are just some of the results that we've been able to associate now with each of those 3 genotypes I mentioned. And again, this is more just to emphasize that we are now really entering into this area of more individualized and personalized medicine that I hope will only get better and only mean better things for patients in the future. So thank you very, very much for your attention. I hope the basic science lesson was not too much. And so if you have any questions, feel free to talk to me at any point. But are we going to take the questions now, or do you want to take the...
Asymptomatic Waldenstroms. Do you see that in all the types, or is there more one type that you see that in the pattern?
Yeah, that's an excellent question. So, so as Dr. Kyle so wonderfully described, is Waldenstroms starts in this. All Waldenstroms starts in this IgM-MGUS phase, which is indolent and is fine. It just sits there. It doesn't cause any problems. And at some point, it transforms. And we have some ideas about why that might happen, but we actually don't know. And we actually, both MYD88 and CXCR4 can be detected in MGUS patients. So they're probably not responsible for what causes this transition from MGUS to full-blown WM, or from asymptomatic WM to more active WM. So this is something that all of us are very interested in. I know, I think Ramon's been working on this a bit, and a number of people throughout the world, including myself. And so maybe next time we all meet up, we'll have a better answer for you. But this time we don't know.
Okay, thank you.